Okay, this is an instructional video to demonstrate how to assemble the PCR reaction for sequenced DNA, the experiment that we're going to be doing this week of September 25th, 2023. Okay, I have some of the ingredients, water, and I find it more convenient to aliquot some of the water to an Eppendorf tube so that it's easier to pipette to these little PCR tubes. And I've also got this primer, P for primer. It's really a pair of primers for the RPOB gene, and then the third ingredient is this master mix. It has the PCR enzyme, thermotolerant DNA polymerase. It's got the nucleotide triphosphates and magnesium needed for the PCR reaction. Probably best to keep all of these things on ice. And then I've got these two little PCR tubes. This is where the magic will happen inside of these tubes. And I've also got a source of the RPOB gene. Each E. coli cell has a copy of this gene, and this is the gene encodes for the polymerase that rifampicin binds to. So there's something different about this gene on each of these plates, or potentially each of these colonies. So I'm going to pick a colony from each plate, and I'm going to document very carefully the serial number, the culture ID from these two plates. So 2396, and then 2397 on my data sheet. And I'll also put this number onto the PCR tubes in case the tubes get mixed up. Then it'll be printed here, 2396, just like that, and 2397. Each student should pick one colony from each of two plates as template for the PCR reaction. So the recipe is 5 microliters of water in each tube. I'm adding that I'm trying to get it to the bottom of the tubes. Water. And then primer. 5 microliters of the primer. Primer is at a concentration of 1 micromolar. So I'm adding 5 picomoles of primer to each reaction. And now to pick the colony, I pick up a pipette tip. I don't use the micropipetter at all, and I just touch the colony, get a few cells on the tip of the tip, and just verify the culture ID one last time, and then I touch the cells just 2 times to the water and I put the water in the primer, and then I move on to my second plate, verify the serial number, touch the cells on one colony, and then touch those cells, just dab them onto and now we have the cells, we have the water for dilution, and we also have the primers, but we need to add this master mix to each of the reactions, and that's 10 microliters. So now we're gonna, it's a 2x reaction mixture, so we need to dial in 10 microliters and add that. And here it's quite important to change the tips. So you can mix the sample by pipetting up and down 1 time if you want to. It's probably not necessary. I just do it for good luck. And now that PCR reaction is assembled, and we're going to close the lids firmly, make sure that they don't pop open during the PCR reaction. So that's assembly of the PCR reaction, and we'll switch to another video for when to demonstrate how to run the PCR machine.